Hello, everyone, and welcome to the Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled NGS, Target-Enrichment Strategies Enable Precision Medicine. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The widespread use of next-gen sequencing is pushing genomic medicine in exciting new directions, in addition to driving overall sequencing costs down. However, upfront costs of sequencing large numbers of samples can still be prohibitive for many research labs. This scenario leads many researchers to search for methods for enriching subsets of the genome before beginning their sequence assays, which can reduce costs and allow them to focus their efforts on genomic regions relevant to their study. Additionally, since these enrichment tactics afford researchers a more focused approach to sequencing smaller genomic regions, it allows them to employ multiplex sequencing strategies in order to improve genomic coverage and overall data quality. Our first speaker today, Marguerite Odenthal, is a research scientist in the Institute of Pathology at the University Hospital of Cologne. Dr. Odenthal will explain her strategies for next-gen sequencing target enrichment of prostate cancer panels for use in clinical research. Dr. Odenthal will be followed by Vikram Devgan, who is head of biological research content at QIAGEN. Dr. Devgan will cover the development and experimental verification workflow and data interpretation associated with the BRCA-A1 and BRCA-A2 gene-targeted next-gen sequencing panel developed by QIAGEN. Before our speakers get started, I want to encourage you to submit questions for our Q&A session at the end of the presentation. We will try to answer as many questions as we can get to. Simply type your question into the Q&A box on the left-hand side of your screen. Okay, so let's get started. Without any more ado, Marguerite, take it away.
Thank you, Jeff, for this kind introduction. Hi, everybody. In the Laboratory of Translational Molecular Pathology at the University Hospital in Cologne, my job is it to test and to establish novel diagnostic approaches for molecular pathology. And therefore, I would like to give you an overview of my recent work concerning NGS, which is the main focus in our lab. Due to the increasing potential of inhibitors targeting certain signal transducers in various signaling pathways, the demands of molecular diagnostics are greatly changing. In order to avoid expensive and patient-burdening therapies before starting the treatment, the molecular status and molecular changes in oncogenic signaling pathways have to be recorded. Here, the present clinical studies to treat gastrointestinal cancer at the University Hospital Cologne are shown. Importantly, each treatment requires the molecular analysis of a special set of signal transducers because certain mutations or amplifications of signal transducing genes affect inhibitors' value or sensitivity. The same is true for lung cancer treatment, which are extensively studied in the Lung Cancer Group Cologne. The pathway which is most targeted by an individual treatment is the EGFR pathway. Tumor cell mutations of the tyrosine kinase encoded by the EGFR exons 19 to 21 predict the sensitivity of the treatment, whereas mutations in the KRAS gene leading to constitutive activation of the KRAS occurring in lung cancer adenocarcinoma with around 30% report that EGFR inhibition will not be efficient. And the panel of inhibitors and strategies is highly increasing. Thus, novel strategies of treatment of other cancer types are applied to lung cancer, and also new drugs are studied. All in all, this requires more intensive diagnostic analysis of the mutation status of a wide set of genes. For these tests, we perform a macrodissection of the tumor area from the formalin fixed tissue, sections on glass slides. The amount of material is very low, and the material is also of very bad quality because we are using a formalin as a fixative. Also, formalin fixation has several advantages. It has also a main disadvantage because it causes covalent linkage of nucleic acids and proteins, and this in turn results in nucleic acid fragmentation during isolation procedures. But also material is very low. Many signal transducers have to be tested for their amplification, translocation, and also for their mutation status. And of course, we would like to test them all by a high throughput technology like NGS, providing multiplex approaches. But we have started to apply NGS for mutation analysis first. In order now to monitor so many gene loci, the question arises, why do we not analyze the whole genome? First, whole genome or exome sequencing requires a lot of technical capacities. Then we need a high IT storage, and also the costs are quite big. And therefore, hotspot targeting is much more preferred. However, in my opinion, one of the most important reasons is when whole genome or exome sequencing is performed, each locus is only analyzed by 30 to 100 reads. However, by hotspot sequencing, the depth of the sequencing is much higher. And this is important when the composition of a DNA population is analyzed and high sensitivity is needed. For example, when somatic mutations in the tumor occur with high background of wild-type alleles of non-tumor cells, for example, inflammatory cells, then we need a high coverage, and that we can have easily by hotspot cancer analysis. In addition, sequencing of the whole genome will provide us with information which is not requested by the clinician and which might then lead to ethical problems. There are two approaches to target the genes of interest: capture hybridization-based methods or multiplex PCR. Because only low material is available, we choose the approach of hotspot target enrichment by multiplex PCR. Multiplex PCR is followed by adapter ligation, various purification steps, and finally by the data interpretation. We use the multiplex PCR approach to establish various cancer-specific primer sets. All this primary work is now going on to be published. As an official cooperation partner of QIAGEN, last year we have first tested the clinical and actionable hotspot panel. These panels cover targets of high clinical interest as mutations in the KRAS gene or the RAF family, the relevant exons of EGFR, but also other genes like KIT, P53, PTGFR, and beta-catenin. Using a reference collective of different tumors and different mutations, pre-analyzed by different technologies, such a high melting curve in combination with Sanger or 454 pyrosequencing revealed 100% concordance with the reference collective. In addition to these panels, the BRCA1/2 panel, therapeutically relevant for treatment of breast and ovarian cancer, which will be later on introduced by Vic, we used also the prostate cancer panel. So both of these panels are tested on a wide range of patients, and the reason of changing to the hotspot cancer panels of QIAGEN was that we have here a high coverage of genes of interest by all amplicons. And I would like to illustrate this with this example. Before using the BRCA1/2 from QIAGEN, we have used a customized panel, here indicated as panel 1, designed early 2040 by another company, which did not totally cover all exons of BRCA1/2, as here shown for exon 11 of BRCA2, indicated in red. And this missing coverage leads to a failure of variant detection. These variants called by the QIAGEN panel are validated by Sanger, but not observed by the panel 1, as you see here. So high coverage and high quality is guaranteed by an overlapping primer design distributed in 4 pools. In order now to represent around 1,500 cells of the tumor area, the DNA input for each assay is 10 nanogram and 40 nanogram in total. There are different methods to quantify DNA using spectrophotometer-based methods. Everything what is absorbed by 260 nanometer wavelengths will be measured. Or using fluorescence-based methods, then all forms of DNA are measured. And because we want to quantify only the DNA which is amplifiable by PCR, we recommend strongly to quantify DNA by quantitative PCR. So here, this is an example of the quantitative PCR by which only the DNA which is accessible for PCR is considered. And genome copy numbers can be easily determined. The result of wrong DNA quantification is a very high background, which is probably due to few molecules carrying formalin artifacts, which are also then amplified and highly overrepresented then in the library. So therefore, we recommend strongly to perform quantitative PCR in order to determine only the DNA which is accessible for multiplex library construction. There are many purification steps in the workflow of library preparation. We are using a robotic system, in particular for high throughput analysis, in order to avoid pipetting and contamination errors, because automatic pipetting is much slower and much more careful for handling the extremely high numbers of amplicons in the library. This slide shows you the amplicons after multiplex PCR purification 1 in comparison to the final purification step 3. When the adapters are ligated, and then the size is increased by the adapter sequence length. But before loading the library on the platform in our lab, we use the MySeq platform, we perform another quality step of the library. We quantify only adapter-ligated DNA molecules using gene-read real-time PCR, and including a lot of controls, and which gives us a very, very good overview what we can load and how many molecules we are loading. After changing this and integration of this quality step, we can ensure high quality of the running, of the sequencing run, and also a standardized loading. In particular, generation of cluster densities of the MySeq is very equal, mostly around 1,200. To give you now an impression of the insights in data interpretation, I would like to refer to our recent test of prostate-mutated genes. We analyzed 110 tumor samples undergoing different therapeutical treatments, and we analyzed here 31 genes relevant in prostate cancer, and they are targeted by 1,837 amplicons. Analysis of 62 data sets revealed 7,557 variants, from which all silent variants were taken off, and also SNPs are not considered, and then hotspot errors were deleted. What are hotspot artifacts? You can recognize them by the variant occurrence in many or even all samples. They occur mostly by a constant frequency, and that is not a frequency which is representing a SNP like 50% or 100%. So this slide summarizes the recognition of hotspot artifacts. And in this slide, I would like to mention that these hotspot artifacts are gene-specific. They are also chemistry-specific, platform-specific, and bioinformatics-specific. So therefore, when we establish a new panel, a reference collective is needed, or many of the mutants have to be validated by an alternative method, as here is shown for a mutant involved in resistance of prostate cancer therapy, and we validated this mutant by Sanger sequencing. This is the quality control, which is then particularly important when the locus is not covered by a high number of reads, as is shown here in this example where the arrow is. Therefore, high coverage is also an important quality issue which we have to consider and which we also analyze. This slide shows you the mean coverage of the prostate-related amplicons, which is around 1,000 reads per amplicon and sample, which is really perfect. I'm at the end now of my overview. Just let me summarize some important issues according to my experience. So hotspot targeting saves capacities and leads to a sensitive detection of variants. In addition, a pre-designed and custom panel guarantee high coverage of the target genes. And quality control steps such as quantification of the input DNA and also the final library quantification are integrated in the QIAGEN gene read workflow and are very valuable for good NGS results. Last but not least, I would like to thank my co-worker, especially Ulrike Keutsch and Claudia Vollbrecht, but also the whole team of Sabine Merkelbach, processing all diagnostics. And I would like to thank the QIAGEN company, which supported us with chemistry, but also with a lot of experience and good advices. Thank you very much for your attention.
Thanks, Marguerite. That was really informative, and I think our audience got a really good sense of how enrichment strategies can improve not only sequence sensitivity, but also the data analysis. So before we move on to Vikram's presentation, I just want to remind everyone once again about submitting questions they might have through the Q&A box on the left-hand side of your screen. Both Marguerite and Vikram will be available to answer your questions after this final presentation. All right, so Vikram, you're up.
Thank you. And thanks a lot, Marguerite, for such a wonderful presentation. What I'm going to do today is I'm going to talk about my experience about next-gen sequencing-based BRCA1 and BRCA2 mutation testing. And before we get into that, I would like to give a legal disclaimer. All the QIAGEN product, which I will discuss during this presentation, they are intended for molecular biology application. These products are not intended for diagnosis or prevention or treatment of the disease. Having said that, what I have done today for today's presentation, I have split this into 4 different segments. So I'm going to start with very brief introduction about BRCA1 and BRCA2 genes and their association in oncology. And then we'll talk about the challenges associated with next-gen sequencing and what QIAGEN has done using its comprehensive sample-to-insight workflow to address those challenges. And the third part, I will introduce a brief case studies talking about performance and application of BRCA1 and BRCA2 genes. And at the end, I will try to summarize everything. So, as we all know, the BRCA1 and BRCA2, they are well-known tumor suppressors. They have DNA repair and transcription regulation function. And if there are mutations in BRCA1 and BRCA2, either got acquired or inherited, the cell can grow as well as divide uncontrollably and may cause a tumor. And if you really look into the stats, there are around 4,000 women in U.S. Who carry the mutation in this BRCA gene. And they are at very high risk for breast cancer as well as ovarian cancer. Now, one can detect this potential risk by doing the molecular genetic testing of BRCA1 and BRCA2 genes. And we all very well know that the knowledge of risk basically allows the better prevention. As far as the prognosis is concerned, the BRCA1 and BRCA2 related cancer prognosis basically depend upon at which stage this cancer has been diagnosed. So this basically shows that the screening of BRCA1 and BRCA2 gene mutations or variants is more of a public health issue. And now, if we have to connect it back again to the biology, we know that the important point here is BRCA1 and BRCA2 gene are tumor suppressor genes, which means that mutation can occur anywhere along the entire length of a gene. And to do now the effective screening, one need a technology to detect these mutation along the entire length of a gene, but also have that technology should have ability to analyze multiple genes as well as multiple samples simultaneously. And in that respect, the targeted next-gen sequencing is a method of choice. So I hope that by now I would have convinced you that BRCA1 and BRCA2 gene mutation testing is more of, or have a more importance for public health, and the targeted next-gen sequencing is a way to address that. So next, I would like to get into the targeted next-gen sequencing workflow. If you really look into that, the targeted DNA sequencing, it's a long workflow which has multiple different steps, and each step has its unique challenge associated with it. So let's say you are working with the FFP samples, and in that case, if you are isolating genomic DNA from FFP samples, the quality and the quantity of isolated DNA is a big challenge. And once you have the DNA, the next step is the real quantification of amplifiable DNA. And it's very simple on that front. It's like garbage in, garbage out. If you won't quantify your DNA after the isolation properly, which can be amplified or which can be enriched during the target enrichment step, you are going to get low yield as well as non-reproducible result at the end. And then in a target enrichment step, it's all about quick turnaround time, low DNA input, as well as limited starting material. And let's not forget about data analysis and data interpretation. These are still the bottleneck for the complete next-gen sequencing workflow. And what QIAGEN has done, we looked into all these different steps of next-gen sequencing workflow as well as their unique challenges, and then developed product to address these challenges as the listed product has been listed over here. Let's take an example of gene-read DNA FFP kit. So we have developed this kit keeping into the consideration to give high yield as well as high quality genomic DNA from FFP samples. And what we have done, we have not only developed these products to address those challenges, we have developed these products as a standardized and integrated fashion, because we believe that the customer should not be spending their time to optimize this NGS workflow. It's QIAGEN's responsibility, and QIAGEN should be doing that. And that's exactly what we have done. We have built all these products in an optimized and integrated fashion so that you can spend your time in designing the experiment and analyzing the results, or basically interpreting those variants which you detect and finding their clinical significance. In this whole workflow, or a battery of products which has been listed here, what I would like to do today is I'm going to talk more about target enrichment step, which is basically our gene-read DNA spec targeted panel. And I will say that this is a kind of a crown jewel in our next-gen sequencing workflow. So if you really look into the DNA-seq targeted panel, which is for target enrichment, so what it's based upon a multiplex PCR-based enrichment technology. And what we have done over here, we have developed primers all along the human genome, so that it will provide you the ability to select or choose any region, any gene, or any hotspot along the human genome for your research. And to provide very high specificity and sensitivity, we have divided these amplicons into the 4 different tubes. So right now we have around 15 different catalog panel, ready-to-use panel available in our market. And they are available for solid tumor testing, such as the clinically relevant tumor panel and tumor actionable mutation panel. Then for hematologic malignancy, tissue-specific cancer panels such as breast, colorectal, liver, lung, ovarian, prostate, gastric, and cardiomyopathy panel. We also have comprehensive panels for carrier testing and cancer predisposition, as well as the comprehensive cancer panel. And recently we have launched the BRCA1 and BRCA2 gene panel. A lot of work has been done or has been put into developing these panels. We have to ensuring that we are including the genes which are clinically and biologically relevant for the NGS research. So what we have done, we have looked into a different reference databases such as Cancer Genome Atlas, National Comprehensive Cancer Network, Cosmic Database, Cancer Genome Census, as well as our proprietary Ingenuity knowledge base. And we picked the genes which has the mutation which are really relevant for the clinically and as well as the biological significance for a specific disease type. And all these panel has gone through a wet lab validation. And when I say wet lab validation, what I mean by that is all these panels has been tested with multiple different samples or multiple different type of samples in QIAGEN to ensure that you are going to get a very high performance for sensitivity, specificity, as well as coverage. But we also understand that one size doesn't fit all, so therefore we also have custom gene panels available for targeted DNA sequencing. We have an online custom builder which you can use to include your gene of interest or a region of interest and build the NGS targeted panel online. So, as I said that in today's talk, I'm going to talk more about BRCA1 and BRCA2 gene panel. So let's have a look on the design and specification of BRCA1 and BRCA2 gene. So if you look into the amplicon design for this panel, what we have done, we have designed the primers in a more or less overlapping session to ensure that we are covering the entire coding region and also the intron and exon junctions. So in total, we have all the amplicons which we have designed here, they all are 150 base pair in size, which will make it really desirable or really ideal for FFP samples, because you know that the DNA isolated for FFP samples generally are degraded and they have average amplicon size of around 150 base pair. And you need only 14 nanogram of DNA input per sample for analyzing BRCA1 and BRCA2 gene. And if you look into the specification, which we have tested from multiple different samples, we can get 98% of specificity as well as 97% of uniformity at 0.2x mean. And most importantly, we can cover all the entire region or the coding region as well as the 20 base pair into an intron and exon junction. And all these bases are callable. Now, just to link the design with the biology, again, I would like to point out that BRCA1 and BRCA2 are tumor suppressor genes. And in that case, each and every base of BRCA1 and BRCA2 gene is important for the cancer research, as mutation can occur in any base of the coding region. And the gene-read DNA-seq BRCA1 panels, it provides you the 100% coverage for the coding region and interaction junctions. And all these bases which we have covered in our panel are callable if you are sequencing deep enough, which will be ranging from 7,000 to 10,000x. So besides this, the most important part of gene-read DNA-seq gene panel is their applicability with not only with all the available sequencers, whether it's from iTorrent or it's from Illumina, but these panels are also compatible with all the different sample types, whether you're working with FFP samples, plasma, liquid biopsy, or single cell. Our gene-read DNA-seq panel works with all the samples, and then you can use the same panel for all different sequencing platforms. Besides providing the workflow, the most important part for BRCA1 BRCA2 panel is its integrated data analysis and user-friendly biological interpretation solution. So BRCA1 and BRCA2 panel comes along with the CLC Cancer Workbench workflow and Ingenuity variant analysis. So these two will help in not only analyzing the data, but also enriching the biologically significant variant. So if you look at the CLC Cancer Workbench, it basically has the QIAGEN gene-read DNA-seq panel analysis plugin in it. And the whole workbench is very easy to use, and it's an end-to-end solution. What you have to do is you have to upload your FASTQ file, the raw data which you receive after the next-gen sequencing, and select which data analysis pipeline you would like to follow. And at the end, it will spit out on its own the QC result as well as all the variant detected in an easy-to-browse visualization manner. So the whole workflow, the QIAGEN workflow from the sample to insight, it takes 4 days, starting from isolating genomic DNA to all the way interpreting the mutations or variants detected in the sample. And now, before going into the case study for performance, I would like to highlight 3 important points for this workflow. First is you need just the 40 nanogram of DNA for this workflow. Basically, by the input DNA is very less, so you can save your precious sample for the other analysis. And it takes only 3 hours for the target enrichment. And in this case, you can save your precious time and have a rapid turnaround for this clinically relevant next-gen sequencing. And third and most important point is you need only 1 library prep per sample. By doing this, you can save your precious money and go and get the more results or more analysis done in a limited budget. Okay, next I would like to share a small case study with you, which we have done to evaluate the performance of BRCA1 and BRCA2 gene panel. What we have done here is we picked 6 previously analyzed samples from breast cancer patients and then enriched them using BRCA1 and BRCA2 gene panel along with the whole QIAGEN workflow, and use the PGM sequencer for analyzing the result. This study has been done in collaboration with Dr. Nicola Normanno from Italy. As expected, the sequencing metrics looks really great. We got 99% specificity and the uniformity of 98% at 0.2x of median coverage. And most importantly, the 100% of the bases are callable in this using a BRCA1 and BRCA2 gene panel. Then we looked into the variant which has already been detected using a Sanger sequencing, and all those variants are detected or called using a QIAGEN gene panel. However, when we compare it with the other panels available in a market, the other panels which are available in the market missed few of the mutations because of uneven and low coverage of the gene. And so basically, this shows that our gene panels outperform than other panels available in our market, and which will allow you to detect not only all the mutations, but since these panels are developed in an integrated fashion with data analysis tools such as CLC Cancer Workbench, you can easily visualize all those variants on a genome browser. And not only you can visualize, but you can also annotate those variants as shown here in this slide. You can clearly see what kind of a variant it is. Is it, as shown in this slide, it's a deletion, which the double T, and then you can find it out how many reads it is, how many reads are there to show the deletion, how many total coverage of that region we have, what is the forward or reverse bias, which gene it is. So you will get a complete annotation of the variant using our CLC Cancer Workbench. And not only, and the next step, you can use Ingenuity variant analysis to enrich biologically or clinically relevant variants. As shown over here, when we analyzed these two genes with one sample, we found around 25 different variants. And when we removed the low confidence variant, we still have 25 different variants. Then you can still remove the common variants from it. And then also you can enrich with the so-called deleterious variants from these. So you have different filters in the Ingenuity variant analysis software, which you can use to filter out the variants which have a biological or clinical significance. As shown over here, with a single sample at the end, we started with 25 different variants, but after applying those filters, we end up with two variants which are deleterious in their effect. And you can do this analysis on a single sample, as well as you can extend it for multiple samples and analyze all the samples together as shown over here, and detect what are the deleterious mutations are present using the filtering system. And Ingenuity variant analysis not only enable you to enrich these variants, but also provide the evidence showing the presence of these deleterious mutation in a publication. So at the end, I would like to summarize my talk by saying that the QIAGEN BRCA1 and BRCA2 gene panel basically deliver 100% coverage of all the bases, and it has been wet lab verified, and to provide outstanding sequencing metrics. You will get 99% specificity as well as 98% uniformity when you're looking at 0.2x of the mean coverage. And most importantly, the BRCA1 and BRCA2 panels have developed as in an integrated manner with our data analysis and biological interpretation tools, which are CLC Cancer Workbench and Ingenuity variant analysis, which enable you to not only find the variants or a high-quality variant, as well as enable you to visualize those variants on a genome browser, but also enable you to enrich biologically or clinically relevant variants. And next, the BRCA1 and BRCA2 panels are basically have developed to detect the mutation in whether it's a somatic or a hereditary mutation in all different sample types, ranging from blood to FFP samples, and these panels are compatible with all different sequencing platforms. And if you would like to get more information about BRCA1 and BRCA2 panels, please visit our NGS microsite, and you can go to that using QIAGENNGS.com. And by this, I would like to thank everyone for attending this webinar. And back to Jeff.
Vikram, thank you very much. That was a great presentation. I think you not only showed us how much hard work went into the development of QIAGEN's BRCA1 and BRCA2 panel, but also how a number of researchers could utilize this panel for their own work. So that was really great. Thanks. I would just like to again thank Vikram and Marguerite for two really fine presentations, but I want to let the audience know not to go anywhere because we're going to start our Q&A session real soon. But before we do, you have one last chance to submit your questions for our speakers, so hurry up and submit them now. So as I'm seeing now, looks like we have a bunch of questions rolling in, so let's begin the Q&A session, and we'll try to get to as many questions as we can. Okay, so our first question is for Marguerite. Marguerite, what is the advantage to perform target enrichment by multiplex PCR versus hybridization-based capture?
First, we have most experience with multiplex PCR, and multiplex PCR is working with very low amounts, so that is already a very big advantage in pathology, and providing also high specificity by the PCR approaches. And in addition, it's a very fast procedure. But the advantage of to use hybridization capture technologies for target enrichment might be to combine even more targets of interest, and then in particular to integrate in one NGS run the target enrichment for mutated, amplified, deleted, and translocated loci. So we can perform maybe a mutation analysis, a translocation analysis, and also a copy number variation analysis. And I think that is also a big advantage, but these methods are not so well established as multiplex PCR, as well, at least in our hands.
Okay, great. The next question is going to be for Vikram. Vikram, what minimum coverage would you recommend in the context of mixed samples, tumor mixed with health or immune cells?
It actually depends upon what kind of percentage of tumor cells you have in your samples. But to keep the thing simple, one can detect as low as 5% mutation frequency at any base in targeted region if one can sequence 1000x mean coverage. And it's possible because QIAGEN gene panels provide very high uniformity during target enrichment steps. So in a very simple way, 5% mutation detection is with 1000x mean coverage.
All right. Thanks, Vic. Appreciate that. Marguerite, you told us that the library construction using automatic processing steps is less prone to contamination. How would you control cross or run-to-run contaminations?
So first, we have in the whole procedure always non-template control, so that is very important. And then second, we change always the barcodes between each run. So we never use identical barcodes from run to run. And then also we use only a special set of barcodes in one run. We analyze the run for all barcodes, also for the barcodes of the run before, so that we can see some vertical contaminations. And then in some panels we have also a looking of polymorphisms for germline polymorphisms. So then we can also see by these polymorphisms if we have some cross contaminations.
Okay.
So that these are our controls.
Okay. Great. Mm-hmm. Thanks, Marguerite. Appreciate that. Next one, Vikram. How effective are QIAGEN's targeted primer-based assays at detecting long insertions, indels, which are in coding exons and in frame, compared to other short-read sequencing technologies?
Okay.
So QIAGEN gene panel for target enrichment, these are meant for detecting somatic mutations, SNPs, as well as copy number variation and indels. So all the 4 different variants or mutations can be detected by gene panels. And when it comes to the indels, anything which is less than 30 base pairs can be detected very effectively using QIAGEN gene panels.
All right. So the next question for Marguerite. Marguerite, how are you able to quantify the DNA, and is the DNA size parameter in the QC?
So we started to confirm the quality of our DNA by microfluidic-based electrophoresis, and to ensure then that we have the right size of the DNA. And but in the last year now, we only quantify the input DNA by quantitative PCR, and we include then in the quantitative PCR amplicons which have the same size as the mean size of the multiplex PCR, which we will use then for target enrichment. And so we can be sure that we target all loci, and that the DNA is not so fragmented that we have that the multiplex PCR will not run very good. So that is a very good quality control. And when we use now the gene read PCR of QIAGEN, then the PCR is already adjusted to the panel which we use later on in the multiplex PCR. So we have here very good quality control.
All right. Well, thanks very much, Marguerite. Appreciate that. And I think with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived for the next 6 months on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues. I'd like to thank Marguerite and Vikram again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to QIAGEN for sponsoring this webinar. So hopefully we'll see you all again at another Gen webinar in the near future. Thanks. Bye.
Okay. Bye-bye.